The goal of the course today, I think, is to cover the field of prebiotic chemistry. Prebiotic chemistry can be considered in a very broad way, could be considered as all the chemistry that happened before there was life, and that is an enormous amount of chemistry. I'm just going to treat the subset of that, which I think is relevant to the origins of life, which is, I think, what we are interested in when we talk about prebiotic chemistry usually. And that will be a little bit atypical compared to some of the other lectures that have been in this series on prebiotic chemistry over the years. If you want to see kind of more of a global view of all the work that's been done in prebiotic chemistry, I would recommend going back onto YouTube and looking for some of the previous lectures. I'll make reference to some of those things, but only briefly. So I think the first thing that we should ask ourselves is, what is the goal of origins of life research, prebiotic chemistry, right? What do we want to achieve with this? And I think one of the things that we want to do is to not just come up with some sort of a scientific version of a creation myth, but actually to gain a deeper understanding of biology than what we have now. And so after his death, the famous physicist Richard Feynman, who was the founder of quantum electrodynamics, it was found scrolled on his blackboard in his office by his colleagues saying, "What I cannot create, I do not understand." And I think that's really true. Until we were able to recreate life in the lab, we can't really say that we completely understand it. And that's my goal in this research. So I'll give you the slide that everyone gives, this rough timeline for the Earth. Not to go into much details because I'm not the expert on this. You've seen much better courses on this than what I'll say. But if the Earth is roughly 4.5 billion years old, we already have some evidence that's disputed for when life, for what we have in the geological record, somewhere between 3.8 and up to 4.2 billion years ago. And that seems to be somewhere where life started. And for that huge period of time after that, for billions of years, life was thought to be unicellular. So we're talking about prokaryotes, the archaea and the bacteria. So all those things that we think about, you know, as my kids think about as ancient, like dinosaurs, that's all very recent things, as you well know. So we're talking about prokaryotes, and there's a concept in this area that has been around for a long time now called the last universal common ancestor, and that is the ancestor to those prokaryotes, to the bacteria and the archaea. And there are some techniques and some studies that have been done to try to date how long ago LUCA, or this last universal common ancestor, lived. It seems to be somewhere between 3.5 and 4.5 billion years ago, depending on which technique you use. So it's constrained in that range. And it seems to have had a metabolism similar to modern microbes. There's a lot of disputes about exactly what those metabolisms were, whether it was completely autotrophic, meaning it makes all its own food, or whether it was actually a heterotroph or a mixotroph. What seems to be solid is that the general organization of metabolism has never changed. It's still based on the same general strategy of how you assemble things and put them together. There's also some debate about what sort of environment this organism, or if maybe it was even a collection of organisms, lived in. There's some people who think it's thermophilic, some people who think it's not, and I don't have any solutions to that, but I just wanted to mention it. And that the reason this analysis is so debated and controversial is because of horizontal gene transfer. So organisms, these cells can just bump into each other and transfer genetic information without reproducing, and that complicates any ability to reconstruct a tree of life. That otherwise it would be much simpler if they reproduce just like we do, right? We could just use 24andMe and we would know the answer. That's not how it works. So I think we should pick that last universal common ancestor, as little as or as much as we know about it, as the target of our origins of life research, to construct something like that from chemistry as the ultimate goal. So the next thing we have to think about then is, what are we trying to reconstruct? And what I'll try to do, and actually spend most of this lecture on, is talking about what life is doing and how that works, so that we have a picture of what it is we're trying to actually explain through prebiotic chemistry. I think this is actually a very, very important issue. And if it's the only thing you take away from this half of the course, it's that we should have in mind what it is we're trying to actually explain. Otherwise, we're doing research that's kind of going in all the directions, and it's questionable whether it's actually teaching us anything about the problem we're trying to solve. So what is life doing? I'll use this analogy. It's doing a lot of different things at the same time. But one of the most fundamental things it's doing is a little bit like a fountain. What is a fountain doing? There's a pump somewhere off screen, it's burning fuel, and it's channeling water through a pipe that points up. That water moves up, it doesn't go into space and dry out the pond because there's also another force acting on it, which is the force of gravity. That gravity brings that water back down into the pond and allows that water to recycle. So we have two opposing forces that are roughly the opposites of each other, both different thermodynamic driving forces operating at the same time, and that is what gives the fountain its dynamic nature. The molecules in the fountain are constantly turning over. They're not the same, but the pattern is what remains constant. And life is very much the same in that regard. So any ecosystem will be taking small geochemicals like carbon dioxide and nitrogen or ammonia or phosphate and converting them into small molecules that we call carboxylic acids or keto acids. And then with the entry of ammonia into metabolism, it converts them into amino acids. With the entry of phosphate, it converts them into sugar phosphates. And those sugar phosphates and amino acids and some of these C1 compounds derived from here can react further to produce the nucleotides and to produce coenzymes. The nucleotides can polymerize under phosphorylation to give the genetic polymers like RNA and DNA, and the amino acids can polymerize also under phosphorylation to give proteins. Now, in this direction, that phosphorylation is driven by the collapse of proton gradients over membranes. That's what produces the phosphorylating agents in biology. ATP is the one that we know about. And it's driven by reducing agents. This is reductive chemistry overall as a net process. At the same time, those things are breaking down and recycling. And that breakdown or catabolism is driven by oxidizing agents coming from the environment and water, hydrolysis by water. So two different forces, both operating at the same time, creating this dynamic build-up and breakdown that's constantly happening in all ecosystems by a very conserved pattern. The molecules are changing all the time, the organisms are changing all the time, the metabolism, what it's doing is remaining constant, just like the genetic code and other core aspects of life. So you can see this even in advanced organisms like us. You can see in mammals, mRNA has a medium lifetime of only 9 hours. It's being continuously synthesized and broken down. Proteins, the same thing. The median protein only lasts for 46 hours. It's broken down. Okay, so now if we're talking about this LUCA, a very simple organism with a very simple metabolism, still very complex, but simple compared to maybe ours today. We have some pathways that fix CO2 and convert them into these carboxylic acids. We have, there's two we'll discuss today called the acetyl-CoA pathway and the reverse Krebs cycle. You have pathways that bring nitrogen into this. This is some of the simplest amino acid biosynthetic pathways. We'll look at those. We're going to look at how phosphorus enters metabolism and produces the sugars, and then how those things come together. I'll briefly go over that to make the nucleotides. So this is, we could consider this the minimal anabolism or build-up pathways of a simple prokaryotic organism such that might have been around roughly 4 billion years ago. Okay, so this is called the Wood-Lungdahl pathway. This is where CO2 hits the road for the first time. Now I should mention there's actually only 6 CO2 fixation pathways that are known. There's a 7th one that's kind of new and debated. It might also be in this list one day. But this is unique, I think, because in 4 billion years, we only managed to find 6 ways to fix CO2, and actually 4 of them are very similar. You can almost consider them like one thing, I would argue. Okay, out of these 6, this one is special, this Wood-Lungdahl pathway, because it's the only one that's a linear sequence of reactions. Like A becomes B, becomes C, becomes D. There's not a whole bunch of cycles or anything like that. It's just a linear path. So it's unique in that way. It's also unique because it's the only one that starts from carbon dioxide only. You don't need any pre-existing organic molecule to build upon. This one just starts from CO2. The other thing that's interesting about it that you should know is that almost every step of it is mediated by transition metals. Metals like molybdenum, tungsten, iron, nickel, and cobalt. And I know many of you are not chemists, so I don't want to spend all this time in all the details of this pathway. So you start off with CO2, and it gets reduced with 2 electrons from the organism. usually on a molybdenum or tungsten cofactor, and that reduces that down to something that's essentially equivalent to formate, right? And it's bound to an organic cofactor. We don't have to worry about all that detail. Eventually, it gets reduced again with another two pairs of electrons to give you something that's equivalent to methanol. It's like a methyl group right here. It's bound to that organic cofactor. And then it gets transferred onto a coenzyme that is a combination of cobalt-1 and iron. So we have now organometallic chemistry. We're forming metal carbon bonds. This is something that you tend to learn in advanced organic chemistry courses in third or fourth year of your undergraduate, but biology is doing this right away in its metabolism. And then there's another branch of this pathway now where carbon dioxide is reduced to carbon monoxide, and that carbon monoxide gets bound to a nickel cofactor. So now we have nickel chemistry. The methyl group on the cobalt gets transferred to the nickel center, and then that carbon monoxide inserts between the existing carbon-nickel bond, and you get a new, what we call acetyl group, okay? And this path, that's equivalent to acetate, and this pathway can then undergo what's called a reductive carboxylation, and that happens on a cofactor that's based on iron, ferredoxin. And so now we've used all the metals. We've used cobalt, we've used nickel, we've used molybdenum and iron to do this chemistry. It's all metal chemistry. And it's all very concentrated early on in this metabolism. Now, there's other cofactors that are involved that are very complicated, and we won't talk about them now. But I want you to appreciate what you should take away from this is that the first place where carbon dioxide gets integrated into metabolism involves all transition metal chemistry with fairly abundant metals, and it's carbon metal bonds, basically. Okay, now, before we get into the next pathway, I want to introduce a concept called autocatalysis. It's a very important concept in chemistry because it's the self-reinforcing feedback mechanism within chemistry. So in the simplest phrasing of it, if you have A, which reacts with B, you get 2B. Then you could subtract B from both sides, and B would be both a product and a catalyst for that reaction. So it's a reaction whose products catalyze itself. So it can speed up as it goes along, and it has an unusual kinetic behavior. It has the potential for exponential growth in chemistry. And there's another way of phrasing this, that if the structure of that reaction is in a reaction network that looks something like this, we can call it network autocatalysis. So now imagine you have molecule A that reacts with some reagents to become molecule B. That reacts with more reagents to become molecule C, more to become D, but now D is big enough that it can split into two and give you back one molecule of A and one molecule of B. That way, every time the cycle turns, you double the number of intermediates you have in that cycle, and you can grow, in principle, exponentially the concentration of those intermediates and privilege them over all the other reaction pathways that might be also occurring, right? So it's a mechanism to funnel energy and matter towards one set of products rather than everything. And so this actual concept's been around for a long time. It's been promoted in the origin of life field for a long time. It's been criticized in the origin of life field. Famously, Leslie Orgel, who was a prominent origin of life chemist, criticized the idea that cycles like this could emerge spontaneously. Because they can bleed off into side reactions, and those side reactions can diminish the efficiency and prevent it from exponentially growing, and actually it could die out. However, he never really talked about the possibility that you could have inputs of those reagents into the cycle, and that this amplifying feature would just help stabilize this linear path through this from A to D. Forget the exponential growth, it would just help this linear path outcompete another perfectly linear path that didn't have this feedback feature. So whether or not you actually have exponential growth or not, to me, is actually not the point. The point is this network structure will help you outcompete something that doesn't have such a structure. So even if it's not perfect. Of course, if it is perfect, then you can get exponential growth. Okay, so the reason that we talk about this a lot in the context of early biochemistry is because of this pathway known as the reverse Krebs cycle. And it is really important because it produces the building blocks of everything else. So I'll just mention the first intermediate you could say on this would be acetyl groups that coming from that Wood-Langdal pathway I mentioned before. And those are the building blocks for all the lipids. Pyruvate, which is the next intermediate, is the building block for all sugars as well as some amino acids. The next intermediate, oxaloacetate, is the building block for amino acids like aspartate, and those go on to make the nucleobases for RNA. Later on, we have succinyl groups, which become incorporated into pherols, which are important cofactors for biology, and ketoglutarate, which is going on to become other amino acids as well. So you really need all of these intermediates. You can actually build all of biology from these 5 intermediates, and they're all in one cycle, and it's the only cycle that has that property. So for that reason, this is really interesting chemistry. If you have but it's also interesting because it's got this autocatalytic feature I mentioned before, which is that you start off with acetyl, which is 2 carbons. You go to oxaloacetate, which is 4. You eventually go to citrate, which is 6. And then that 6 breaks up and back into the 2-carbon acetyl-CoA and the 4-carbon oxaloacetate. So you've got this autocatalytic feature, or network autocatalytic feature in this cycle that gives you the building blocks of everything, right? So this attracted lots of attention from people interested in the origins of life as a potential pathway for getting life started. Now, it's not the only pathway. Well, I also mentioned there's not a whole bunch of different mechanisms in this. I've color-coded them down here, and you'll see there's a lot of redundancy in the mechanisms. There's a reaction here in blue. It's called reductive carboxylation, but it also happens again here. Here you have a carboxylation, so you add CO2, it happens again here. Here you have a reduction, it happens again here and here. Here you have an addition or a subtraction of water, that happens here, here, or there. So you don't need that many mechanisms to make this cycle happen. So people also speculated it may be a result of self-organized processes and not of evolutionary biology. All right, I wanted to mention something that I don't think gets said enough in this field. It's about the energetics of the reactions involved. You may have noticed on the previous slide that I brought up, there was these types of molecules called thioesters that appeared as intermediates in that cycle. And there's some things I wanted to say about that in terms of the energy, because those thioesters get formed by starting with the carboxylic acid and using ATP to phosphorylate the carboxylic acid, usually to something called an acyl phosphate. And then the acyl phosphate reacts with another molecule that's based on sulfur called a thiol, and it forms this thioester. That is a step that happens a few times in that cycle, and it happens all over biochemistry, in fact, as you'll see in the next slides. But there's something very interesting to me about it. It's that if you look at how much energy there is in ATP when it donates a phosphoryl group, there's a kind of an indicator of that energy that we call the heat of hydrolysis. It's how much energy is released when water hydrolyzes that substrate and gives you back phosphate plus the hydrolyzed byproduct. And for ATP, that energy is minus 30 kilojoules per mole. So that's okay, nice. But when you look at the energy of the acyl phosphate, it is minus 43 kilojoules per mole. So it's actually, you release more energy by hydrolyzing the acyl phosphate than you do ATP, which means that ATP, phosphorylating a carboxylic acid to an acyl phosphate, is energetically uphill by 11, roughly, kilojoules per mole. Now that's, of course, an analysis where it's a one-to-one reaction. So why would your energy currency, ATP, be doing a reaction that's thermodynamically unfavorable? And why does it work at all then, if that's the way it works? Well, the answer is it doesn't occur in a one-to-one ratio. The reason that that reaction can go uphill is because the cell maintains an ATP to ADP ratio. ADP is the byproduct of hydrolysis of ATP. It keeps that ratio at radically different ratios than it should be at equilibrium. So it's, for those of you who have heard of Le Chatelier's principle, if you have lots of one reagent and large excess, you can push a reaction towards the other side. That is actually what's pushing these reactions around. It's not the energy inherent in the strong PO bond that people often talk about. They always call it a high energy PO bond. It's actually not high energy compared to what it's trying to make. Okay, so this is a very important point. So what we have to be thinking about, what's driving these reactions, is not the chemical bond energy of ATP per se, but the incredibly out of equilibrium ratio of ATP to ADP that the cell maintains through a process called chemiosmosis that we're going to look at later. And that's really important in understanding how the cell works. Okay, so then once you have this acyl phosphate, to convert it to the thioester, the heat of hydrolysis of a thioester is minus 35 kilojoules per mole, give or take. So it's slightly energetically downhill to go from the acyl phosphate to the thioester, but not much. So these are higher energy intermediates than the carboxylic acid and ATP when considered on a 1-to-1 ratio. So you have to ask the question, why would life ever use an energy currency that's so weak? Why not use one that's strong, that makes the reaction push to products automatically? We can speculate, but we don't actually know. I'll give you a suggestion at the end, further on in the lecture, of why that is, probably in the next half. Okay. All right, so I talked about the reverse Krebs cycle, this cycle that makes everything that's exponential. And I mentioned that there's 6 total carbon fixation pathways, one of which is that linear one, and then there's 4 that are very similar. And those 4 include the reverse Krebs cycle that I just showed you, but 3 others as well. And here's what they're called, I don't even know how to pronounce them, dicarboxylate 4-hydroxybutyrate cycle, the 3-hydroxypropanate cycle, and the 3-hydroxypropanate 4-hydroxybutyrate cycle. Now, we're not going to go into look at their chemistry, but what Brockman and Smith have done is they've made like a network map of these reactions, and they've overlapped them so you can see how they overlap. And you can see that what they're all doing, they're all network autocatalytic, all 4 of them, and they either directly produce or indirectly through just a couple of reactions produce those 5 universal building blocks that I mentioned before. So essentially all 4 of these cycles have a very similar function. They have an exponential growth mechanism built in to build the keto acids, and they all do that one way or another. And they propose that these were evolved or selected for because they each functioned optimally under different conditions. So an organism living under one condition might choose one rather than the other because this works better under cold conditions or alkaline conditions, etc. So really, there's actually very little diversity. If you consider all 4 of these to be the same thing, there's actually very little diversity in how life fixes CO2 into its metabolism. Okay, how does ammonia, nitrogen, that's another element. Okay, how does that enter metabolism? Well, it enters through a reaction known as reductive amination. It enters always in the same place. And it starts with the metabolite ketoglutarate. And you have ammonia plus 2 electrons come in, and it gives you glutamate, glutamic acid. And once it also enters the second time from glutamic acid, you can also use ATP to activate this carboxylate and then substitute it with ammonia to give a terminal amide, which gives you the amino acid glutamine. So these are the 2 places where ammonia comes into metabolism, and everything else is reshuffled from there. Okay, pretty easy. All right, ammonia comes in here, and then once it comes in, it gets reshuffled through a process known as transamination. So once you have your glutamic acid, it can go to the other keto acids that are around. It can pass that ammonia to them, and it'll convert, let's say, pyruvate into alanine. It'll convert oxaloacetate into aspartic acid. It'll convert glyoxylate into glycine. There are other ways to make glycine besides this. This is just one of the ways. But these 2 amino acids are very important because they're the ones whose carbon skeletons end up in the nucleobases of the RNA. So if you want to make your genes, you have to have those amino acids. All right, so how does this actually happen, this process of transamination? Well, once you have your glutamic acid, or it will shuttle its ammonia to a small organic molecule, which is also produced by metabolism, called pyridoxal phosphate. And so it'll bring that ammonia and 2 electrons to pyridoxal phosphate. It becomes something called pyridoxamine phosphate. This is vitamin B6. You take this when you take your vitamin pill or a small derivative of it. And then that reshuffles that ammonia and 2 electrons into a new keto acid producing the amino acid. Now, you might wonder if ammonia could enter metabolism through reductive amination, why doesn't it just use reductive amination on all the other keto acids? Why shuffle it into one and then reshuffle it through transamination? We don't know the answer to that question. We've tried to probe that a little bit through our studies. And there's also a paradox about all this. You also need this transaminase enzyme for this to work. The cofactor doesn't work on its own. But PLP, this cofactor, pyridoxal phosphate, is biosynthesized using transamination, which depends on PLP. So that tells you that it needs itself to exist. So at some point it must have been produced without itself, right? There had to be a non-enzymatic or non-coenzyme dependent process that made this chemistry happen for this to emerge in the first place. And same is true with the transaminase. The enzyme is produced by metabolism, which is coming from transamination, which depends on PLP, which depends on transaminase. So you'll see that metabolism in life is full of these processes where it's like a snake biting its own tail. You have to start the process somewhere, otherwise it's always looping back onto itself. I could give you a million examples of that. All right, so now we've got pyruvate on our reverse Krebs cycle or from our acetyl-CoA pathway. Now we want to make sugars. The chemistry wants to make sugars, and it does it through a very complex pathway. Two major channels. One is called gluconeogenesis that starts from pyruvate, and then another one called the pentose phosphate pathway. And these are very, ancient and very conserved processes. There doesn't seem to be any alternative to making sugars besides these processes. And I won't, go through all of the chemistry, but it starts off at pyruvate, which comes from these two carbon-fixing pathways. It gets phosphorylated by ATP in a kind of unusual way. Instead of giving one phosphoryl group, it actually gives two stuck together, a pyrophosphoryl group, and it produces phosphoenol pyruvate. And then this phosphate's really important. It gets stuck in the backbone of all sugars and even in RNA, as you'll see. So you don't have to worry about all these reactions. There are things like adding water to this CC bond, an internal transfer of this phosphate group from the C2 carbon to the C3 carbon, and then reduction, phosphorylation of this carboxylate to the acyl phosphate and then to the thioester, reducing it to an aldehyde, and then something called an aldol reaction that basically takes two 3-carbon groups and couples them together to make a 6-carbon group. So it links those two fragments together. That gives us fructose. Maybe you've heard of high fructose corn syrup. Okay, this is how this is produced in biochemistry. And then there's a hydrolysis of a phosphate group to give you a free alcohol, and that's how you get glucose. And so from glucose 6-phosphate, then you can move over here, and you start into this other pathway called the pentose phosphate pathway. And so that involves a few more reactions to chop off a carbon, releasing CO2 and giving you 5-carbon sugars. That's the ribose family, okay? So that you have ribulose 5-phosphate, and then you get from this ribose 5-phosphate. Now that's really important because once you phosphorylate this thing, it becomes phosphoribosal pyrophosphate, or PRPP. It's a 5-carbon sugar, and this is the building block of all nucleotides. Both the pyrimidines and the purines, they come from this one intermediate. Something very special about this intermediate, and I think that needs to be investigated more, why it's so special. So there you go. Now, of course, this stuff also happens in reverse, right? A lot of these pathways, they run also roughly in reverse, not the microscopic reverse. They're actually driven by oxidants, so it's a different thermodynamic driving force, and that pathway is called glycolysis. So you've probably heard of that too. We're running glycolysis now as we digest our breakfast. We're also running the oxidative Krebs cycle, not the reductive one that I mentioned in the course. But we think those, well, maybe not glycolysis, but at least the oxidative Krebs cycle probably was not that dominant until there was more oxygen in the atmosphere. But that is debated, actually, so it may or may not be true. Okay, I debated, normally in this course, I have a slide on the actual purine and pyrimidine biosynthetic pathways. I decided that since most of you are not chemists, that probably that's overkill. So I took it out, but I just wanted to give you an overview of how these processes work. Okay, and I've color-coded it. So here are the purines, and you can see where every atom comes from. Okay, this NCC here comes from glycine. This nitrogen comes from the nitrogen in glutamine. So it could also come from ammonia, right? Ultimately, because this comes from ammonia. And this carbon comes from a C1, it's basically a formate derivative, either formal phosphate, which is very simple, or a more complex organic fragment, but the formal group is what's important here. So it's very simple C1 chemistry. This one comes from bicarbonate through phosphorylation, and this ammonia comes from the nitrogen in aspartic acid. So It's all very simple central metabolites that are used to build these nucleotides. The pyrimidines, same thing. The nitrogen here comes from carbamoyl phosphate, which comes from adding ammonia into CO2 plus ATP, basically. Okay, and this fragment here comes from aspartic acid, another simple amino acid. This carbon from bicarbonate, and there you go. You've got the nitrogen here coming ultimately from glutamine. So with just the metabolites that come right off this reverse Krebs cycle through transamination and through the, you get everything you need to make all the nucleotides. You don't need a huge metabolism to be able to make these sorts of things. Just a random observation that I've noticed over the years is that every amino acid that you need to do this, for some reason, if you look at its codon in the genetic code table, it always starts with G in every case. Coincidence? Maybe. Okay. Now, I wanted to just mention this point because it's also something that I don't ever see brought up, even in the literature, about anabolism. And if you follow all the reactions that I either showed you or was going to show you in these pyrimidine and purine biosynthetic pathways, you'll find that even though I told you that overall they're reductive, and that's true, there's still some oxidative reactions. There's 4, in fact, through all those pathways. Everything from CO2 to nucleotides. And I've written them out here. But what you notice is that these 4 oxidative reactions, now oxidation formally is just the having something accept electrons, right? So it doesn't mean you need oxidative conditions per se. You just need an electron acceptor. And if you go to Equilibrator, which is like a program you can use to estimate the thermodynamics of metabolic reactions under different circumstances, you can find that all 4 of these oxidation reactions are actually thermodynamically favorable even under mildly reductive conditions, actually moderately reductive conditions. You can go to minus 300 millivolts and most of them are still thermodynamically favorable, even though they're oxidations. So that tells you these are relatively easy oxidations, at least from a thermodynamic standpoint. So they could still happen under reductive conditions. You just need something to take the electrons off your hands. And that's why organisms can live at hydrothermal vents, for instance. Otherwise, they couldn't. Right? Okay, so I've tried to summarize, because you're not all chemists, I wanted to summarize what we've learned so far. So metabolism has an overall structure and a way of doing things that has remained conserved since as far back as we can see using genes, roughly 4 billion years. Redox reactions in anabolism are nearly all reductive, except for 4, which are oxidative, but those are relatively easy even under reductive conditions. Core metabolism, going all the way from what I'm considering that, going from carbon dioxide to nucleotides, uses a small set of reaction mechanisms that repeat over and over again. There's not that many one-off mechanisms, and when they do happen, I think it's very special, and we can learn from them. There's all 4 carbon fixation pathways that make the same keto acids have a built-in self-amplifying network structure called network autocatalysis. I've shown you that the phosphoryl groups are a key structural component of the primary metabolites, like the sugars, phosphates, and that carboxylate phosphorylation, in particular, which has this relatively large heat of hydrolysis, is a key repeating mechanism in anabolism, and it's actually a better phosphoryl donor than ATP, which actually produces it. And the reason that that can actually happen with ATP is because of this really high and out of equilibrium ATP to ATP ratio. It's not the strength of the PO bond that drives phosphorylation, it's this out of equilibrium ratio. So that's what I think you should remember from this. And I will give you overall in the 2 courses 6 slides like this, and then I'll condense them all down into 1 slide. So, so if you can't remember anything, you can just remember that 1 slide. Okay. So how does this ATP to ADP ratio come about? Where does it come from? Why? It's so important, obviously. So where does it come from? It comes from, a very important enzyme called ATP synthase. And what ATP synthase does is it happens to be embedded in a membrane. On the outside of that membrane, on the outside the cell, I'm talking about prokaryotes here, not in a mitochondria. We're talking about, simple organisms. Okay, they have a pH gradient across that membrane. Now, what does that mean? It means that on one side of the membrane, the pH is 3 units lower than it is on the inside. And so naturally, by diffusion or osmosis, those protons want to come back into the cell. When they come back into the cell, they pass through ATP synthase, and it couples the re-entry of those protons into the cell to gluing ADP, this is the dephosphorylated form of ATP, plus phosphate together to produce ATP plus water. So it's taking this proton gradient, and as it collapses and goes back to equilibrium, it forces ADP and phosphate together to give you ATP. And this process is responsible for roughly 80% of the ATP production in prokaryotic cells. There's also some ATP made by substrate level phosphorylation, but this is the main mechanism by which it happens. And this thing is also universally conserved. Okay, so that's how it comes from. That's how we get this very far from equilibrium ATP to ATP ratio that powers all of metabolism. So that's probably a pretty important thing that we'd like to explain when we want to understand the origin of life. Why is it that way? Now, if you look at this from a physicist's standpoint, when you have a proton gradient, that's the language biochemists like to use, right? But what is a proton gradient? It means there's more of one charge on one side than another. So you have charge separation. When you have charge separation, you have an electric field, right? So you can't really separate the idea of a charge gradient and electric fields. And there's very strong electric fields in biochemistry. You'd be kind of surprised maybe how strong they are. So if you just look across the membrane of a prokaryotic cell, you typically find an electric field strength of 10 to the 7 volts per meter. Okay? Think about a 9-volt battery. This is 10 to the 7 volts per meter. It's like a bolt of lightning, right? But the actual potential is not that strong. The potential across that membrane is only about 70 millivolts, 0.07 volts, right? So that's way weaker than a 9-volt battery. The reason it's so strong in terms of its electric field is because the membrane is only 5 nanometers big. So you have, you can estimate the field strength from the voltage drop, or the potential, divided by the distance, right? So you can get a big field strength either by having a very strong potential or by having a very small distance. And this does the second way, right? So this is probably also really important. If we want to understand life and explain life, why is it that for 4 billion years, prokaryotic cells have been stuck on this very small size regime of 0.1 to 10 microns, roughly? Okay, there's some counterexamples, but the vast majority fall within that window. Why is that? It's probably very important. And so people have recently started to realize that actually this gradient is also linked to the fields, and so that now there's some simulations of ATP synthase and how the electric field that is produced influences its motion. So maybe we've been thinking a little bit too much about the proton gradient and not enough about the electric fields, and this can also explain why this 3-unit pH gradient is able to produce ATP and do its work. All right, so where does that proton gradient come from? Nature doesn't provide it for you. So where does it come from? The cell makes it itself. Okay, it makes it itself through using the electron transport chain. So what happens here is as metabolism happens and electrons are kind of stuck onto its coenzymes, they get transferred back to the membrane through a series of proteins that are all kind of electronically connected. And that electron can hop from one to the other. And as it does, it goes to lower and lower potentials, so it gets more and more stable. And that energy gets transferred into a pump that pumps protons out of the cell outside. So the cell spends a lot of effort through its metabolism. It uses the exergonic nature of its metabolism to pump protons out of the cell, and then it uses that gradient to reform ATP. So it's basically converting electrical energy into phosphorylation energy or dehydrative energy. This is this mechanism by which it does. So one can think evolutionary, how did this system evolve, right? You had to have the gradient to get the to make ATP in the first place, but you had to have metabolism running to make the gradient. And so there's got to be some solution to that chicken and egg problem as well. But very interesting. Okay, another thing I wanted to mention about, I mentioned the coenzyme PLP already, right? And lots of other ones. This is vitamin B6 that I mentioned, but there's many other ones that are important. There's NAD, which is existing in 2 forms, reduced NADH and oxidized NAD+. That's extremely important for metabolism. Other ones like FAD are also important. They're summarized in this slide. You don't need to look at the structures per se, but I did want to mention that many of them contain ADP as a unit in their structure, especially NADH. It has that. And I think you could argue ADP itself is a coenzyme because it shuttles phosphoryl groups. And about 30% of all enzymes use coenzymes to allow their chemistry to happen. So one can ask the question, why do we have coenzymes at all? Why don't enzymes just catalyze everything on their own? And that's something we have to consider when we think about life's origins as well. Now, if you actually, these are all made by metabolism itself, and they allow the reactions of metabolism to occur, even their own synthesis. So there has to be chicken and egg loops here as well. And so Andreas Kirschning in Germany has gone through the biosynthetic pathways of all of these cofactors, and he's thought about what existing cofactors need to already exist to produce it, and he's mapped out what he thinks are kind of like a order of appearance of these cofactors over evolutionary time. And so they probably all appeared relatively early, but they probably came in this order according to this analysis. I haven't really gone through all of these things in detail to see if I agree with them, but I generally think it looks realistic. PLP is one of the earliest ones. It's used right away in metabolism. NAD is another early one. FAD as well. These are CoA. They all appear relatively early, and more sophisticated ones that need more complex metabolites occur later. That seems to make sense. So they are very important for metabolism. They're produced by metabolism, and some of them appeared before others. It's what you should learn, and you should wonder why they exist at all and why they're important in biology, and that has to factor into your origin of life thoughts. Okay, so next take-home message slide. Most ATP, roughly 80%, is produced by ATP synthase, which uses the collapse of proton gradients across membranes and the associated electric field to condense ADP plus phosphate into ATP plus water. It's producing dehydration chemistry. Then the pH is kept lower outside the cell by the electron transport chain, which pumps out protons by coupling exergonic electron transfer to a terminal oxidant. Nearly all prokaryotic cells have remained in the size regime of 0.1 to 10 microns as far back as we can infer. Coenzymes are small molecules that can co-catalyze roughly 30% of enzymatic reactions, and some are more ancient than others. Okay. All right, let's move on. Now we're going into the nucleotides and proteins. Okay, and I think we're on track. We're doing well. Okay, maybe I'm even ahead of schedule actually. So RNA and DNA synthesis is catalyzed by proteins. That's what I want you to remember. So there's a very specific way in which RNA is synthesized from the nucleotides, whose pathways I didn't show you in detail, but I summarized in that one slide. Once in the biosynthesis of these nucleotides, you initially get what we call the monophosphate. So if this is the sugar part and this is the nucleobase part, there's also this phosphate part, right, the chain, and you either have 1, 2, or 3 phosphates. Now in the original biosynthesis of the nucleotide, you get always the monophosphate originally, and then you phosphorylate one at a time to get the diphosphate and then the triphosphate. It is the triphosphate that gets polymerized into the polymer, RNA or DNA. But the synthesis of DNA specifically always comes from the ribonucleotide. Okay, so you start off with the ribonucleotide diphosphate, so with 2 phosphates. The hydroxyl group at the 2' position gets reduced, and you get the deoxynucleotide diphosphate, and then it gets phosphorylated to the triphosphate before being polymerized into DNA. So there's very specific ways, for some reason, where these chemistries happen and how they get incorporated into the polymer. So that's just something you should know. It's the same in all life. But that polymerization process is catalyzed by proteins. So you need proteins to polymerize the nucleotides into the genetic polymers. Okay, and that's kind of the main message I wanted to send. It's not to say that it can't be done other ways, but this is how biology works, right? So how about proteins? Well, they are catalyzed by RNA. Okay, so you have the ribosome that is at multiple levels. Okay, so I'll show you what I mean. So here's your amino acid that you've now produced by your biosynthesis. It reacts with ATP to give you an intermediate called the aminoadenylate, okay? The aminoacyl adenylate. So it's basically the amino acid group as an anhydride with the monophosphate form of, so it's AMP, adenosine monophosphate here. Now, why is it adenosine monophosphate, not one of the other nucleobases? We don't know. Why is it the monophosphate chain rather than a diphosphate chain? We don't know. But the important part is already here you have a ribonucleotide attached to an amino acid, even at the monomer stage. And then this can get incorporated into, picked up by tRNA, which is a chunk of RNA, and it shuttles it over now and brings it to the ribosome and places that on the growing peptide chain. And the ribosome itself is mostly RNA. There's some peptides attached to it too and stuff. But so you need proteins to synthesize the genetic polymers, like RNA especially, and you need RNA to synthesize the proteins. So another chicken and egg loop. Okay, that's what I want you to recall. RNA does not synthesize RNA. Sorry, it doesn't do that in biology. Yet it's at the center of many origin of life hypotheses, which what I'm showing you this for is maybe to question that that hypothesis is actually true or not. It may be, but there's alternatives to that. Okay, so the next thing I wanted to deal with is how do enzymes actually work? Okay, how do they work? This is actually surprisingly hard to probe. We have many crystal structures, X-ray crystallography structures of substrates inside their enzymes, but that doesn't tell you how the catalysis actually happens. It might give you a snapshot of what the active site of this enzyme looks like, but that doesn't tell us how it works exactly. And it's been very hard to probe that because it's so complex. So there have been different ideas about it, and before I can introduce one of them, I need to explain a fundamental concept about how electric fields influence chemistry. And so this is actually a relatively recent development in the field of pure chemistry. It comes from predominantly the lab of Sasson Shaikh in Israel, and he's interested in studying how man-made electric fields influence a chemical reaction, both theoretically and others that he collaborates with experimentally. And the theory is this: if you have a reactant that has a certain dipole moment, okay, so how, where that electron density exists within that molecule on average, it will react to give you the product. Now, in reacting, every chemical reaction has to go through a kinetic barrier to get to the product. That's good, otherwise you'd spontaneously combust because you'd react with oxygen. Right? But every reaction has a kinetic barrier, and you have to get over that kinetic barrier to get to the product. The highest point on that pathway over the kinetic barrier is known as the transition state in chemistry. And when that transition state also has a dipole moment associated with it. Now, if the dipole moment of that transition state is larger in magnitude than the dipole moment of the starting material, then if you apply an electric field, you will stabilize that transition state more than you stabilize the ground state. And if you do that, relatively speaking, you lower the barrier to the reaction. So it becomes easier to go over that barrier, and the reaction becomes faster. So you can catalyze a reaction that has a large dipole moment in its transition state simply by applying an electric field. Now, the electric field has to be aligned with that dipole moment, but if the starting material has a similar orientation of its dipole moment as the transition state, the molecule itself will align to that field anyway. So it's not even an issue. Okay? Now, there's also reactions that have a change in the direction of their dipole moment in the transition state, not of the magnitude. And this is an example here. And they can also be catalyzed by electric fields if the field is pre-aligned with the transition state rather than the starting material. Because then you'll stabilize this transition state more than you stabilize the starting material, and you lower the barrier. Okay? And that is something that enzymes do really well, actually. It turns out they apply dipole moments to their transition states very accurately by pre-organizing everything. So this is actually turns out to be very important for enzyme catalysis, and it's also been explored directly experimentally in the field of physical chemistry by applying electric fields on the nanometer and angstrom scale. So this is actually an emerging area of chemistry, but you can imagine all those sorts of things this would change. You can also change equilibrium constants with electric fields. So imagine water. If you have a bunch of water, water will randomly split into hydronium ion and hydroxide ion. That's called the autodissociation of water. But imagine you apply an electric field now. It's way easier to autodissociate to your ions because you've stabilized them relative to the starting material through the electric field. So electric fields can change a lot of things in chemistry. Okay, so why is that relevant? Why am I teaching you about electric fields? It turns out this is probably the main principle of enzyme catalysis. This is how enzymes work, okay? And this was predicted actually a long time ago by a physical organic chemist named Bill Jenks. He wrote a famous textbook where he talked about electric field stabilization or destabilization of the starting materials. It's called the Searcy effect. Turns out that's probably not what was actually happening. There was many theoretical models from Arya Warshel that showed, yes, what we call electrostatics or electric fields do stabilize or do facilitate reactions, but they do so by stabilizing the transition state, not by destabilizing the starting material. Okay, this was, so Jenks had it a bit wrong, but the general idea of electric fields and electrostatics was correct. So Warshel showed in many theoretical studies that actually what is happening, the enzyme is applying electric fields through its hydrogen bond networks on a very, very small distance scale. So it doesn't make large potentials, but it applies them over very small distance scales. And that works for the reasons I just explained in the last slide. Now, the magnitude that, so he did all these calculations, but nobody believes calculations until they see an experiment. And experiments are really hard to do because this enzyme only generates these electric fields on the nanometer, on the nanosecond timescale. And so how could you easily measure it? And so there's some nice work that was done by Steve Boxer relatively recently. He's written a review just 8 years ago now on this. And he was actually able to use spectroscopy on a very small timescale to stare into the active site of the enzyme and see how strong the electric field was. It's something called the vibrational Stark effect. And when you look at this, he can find that there's actually electric fields in enzyme active sites of 10 to the 9 to 10 to the 10 volts per meter. This is 3 orders of magnitude larger than that huge electric field that already exists over the membrane. But this exists because these are over angstrom distance scales now, 0.1 nanometers, okay? So this turns out to be very important for how enzymes work. Boxer actually did a brilliant experiment. What he did is he made a whole bunch of mutants of enzymes, measured the rate of their catalysis, measured the strength of their electric field, and then correlated them. He plotted them. And from there, you can extrapolate back to the intercept and see how strong is this field, how fast is the reaction when there's no field? And the answer is, in most cases, 0, okay? So there's some enzymes that had up to 30% still reaction rate preserved, but most of them were 0. And so Warshel seems to be right. It's electrostatic stabilization of the transition state that is the main principle of most enzymatic catalysis, except for the enzymes that use these transition metals that do organometallic bonds. Okay, so you got both. You got this organometallic chemistry, you got this electric field catalysis, depending on the enzyme. Now, this effect was especially pronounced for phosphoryl group transfer, which is at the heart of metabolism. So maybe electric fields could be important to the origin of life more so than we've thought. They've almost never mentioned in this literature. So the take-home message 4 is RNA is biosynthesized by polymerization of triphosphorylated nucleotides, which is catalyzed by enzymes. Metabolism is catalyzed by proteins, which are enzymes, often with the help of metal ions and coenzymes, but proteins result from the ATP-driven dehydrative polymerization of amino acids, which is catalyzed by RNA, which is the ribosome. Okay, enzymes mostly work by creating a strong 10 to the 9, 10 to the 10 volt per meter electric field in the active site of enzymes, with the exception of some of these enzymes that are based on organometallic chemistry, although they probably also have strong electric fields. I don't think they've been studied in much detail. And metabolic reactions actively involving phosphoryl group transfer are especially susceptible to enzyme catalysis. Okay, we're getting there. We're getting there. Stick in there. Okay, now we're getting to really high level stuff. Redundancy in the genetic code. Okay, so here's the genetic code. For the code involves 3 sequential nucleotides, the first position, second position, third position. The first position is represented here, second is represented here, third is represented here, and the amino acid it codes for is represented here. And what you'll notice by staring at this chart for a few minutes, even a few seconds, is that there's much more redundancy for the amino acid at the third position than there is for at the first or second position. So for instance, if you want proline, you need to have C as the first letter of your codon, the C is the second letter, but U, C, A, or G all give you proline at the third position. So it's redundancy. Okay? Same thing is true for alanine. You need G at the first letter, you need C at the second letter, but if you have T, C, A, or G at the third position, you still get alanine. And this is largely redundant. We call this the wobble position because it doesn't really matter what you put in. It's still, there's an error margin there. Okay? So that's interesting. And because of that, some people have speculated that really it's the first two nucleotides that are important in the codon. And actually you could rewrite the genetic code not as a 3-letter codon, but as a 2-letter one. Maybe excluding some of these. You'll notice some of the more recent amino acids, the more biosynthetically complex ones anyway, have less redundancy, and they have at the third position things like tryptophan or you know, they tend to have, or methionine, they tend to have just one or two codons at the third position that code for them. So the idea is that some of the more recent amino acids have kind of occupied positions that used to be completely redundant. And so, you know, so you can redraw that nucleotide table. And I'm taking this chart from, well, I've redrawn it from a paper in 2005. But this idea dates back to 1975 in a paper from Wang in PNAS, where he noticed there's actually very tight relationships between the first two letters of the codon and the biosynthetic pathway used to make the amino acid. So, for instance, if the first letter of your codon is G, then it turns out that in this two-position code table, the amino acids it codes for are all derived from transamination or reductive amination of the keto acid in one step. Here they are. Okay, these are the ones that came right off the Krebs cycle for the most part. Now, if the first position is C, then they're derived from ketoglutarate through a common biosynthetic pathway. If the first position is A, they're derived from oxaloacetate, and if the first position is U, they're derived from pyruvate. And the second position tells you what type of reaction you do on them. Okay, so if you do, you'll notice that a lot of, if the second letter is C, then you see a lot of them are, you have to activate that terminal carboxylate group and either reduce it, cyclize it, or reduce it, etc. If the second letter is A, then you end up with always a terminal amide at the end by activating that carboxylate. So there's very tight correlations between the first two letters of the code and what reactions are used to actually produce that amino acid in biosynthesis. To me, it looks, and to the people that wrote this paper, it looks that that correlation is tighter than the physical properties of those amino acids themselves, which is the typical explanation given to the codon assignments. So, you know, why is this relationship existing, or this apparent relationship existing? It was speculated upon in this paper. And basically, well, this is a scheme from a review that my lab wrote in 2020, where we've kind of redrawn that in a different way. We draw the actual metabolic map, we draw which amino acids are derived from it, and we number the number of biosynthetic steps used to make those amino acids from the Krebs cycle. And you can see that the ones that start with G have very low numbers, the ones that start with C have very low numbers. The ones that, but the more complex ones that have less redundancy, like these ones, have huge numbers. 27 steps to make tryptophan, it only has low redundancy. So there seems to be some sort of relationship between how early that amino acid is made off the Krebs cycle and what its codon is and how many steps it takes to make it. Now, why is that? Well, Copley speculates that this is because there's some sort of catalytic relationship between amino acids and their codons. That somehow the short nucleotide chains actually catalyze amino acids biosynthesis and become associated with the nucleotides and link to them, and that's what creates these relationships early on. And now we see it through supramolecular interactions between the RNA chain and the amino acid, but it started off in this catalysis way. So, you know, maybe that's true, maybe it's not, but it's an interesting thing to think about. If it's true, though, it requires individual small nucleotides or small chains of them, dinucleotides or maybe trinucleotides, to do catalysis, which, is there evidence for that? We'll see later. Okay, so take-home message 5 is that the association of amino acids and their codons with tRNA is not random. There's a strong correlation between the first 2 nucleotides of a codon and the sequence of biosynthetic reactions used to make that amino acid. Whether that relationship is real or it's just appears to be there is to be determined. Okay, so this is where I was originally hoping to end off today. I'm a bit ahead of schedule, so I might go a couple of slides in to the next part. But to summarize all of those 6 take-home message, or all of those 5 take-home message slides into one slide, what is life doing? This is what you should remember, the stuff on this one slide. Complex but conserved reaction networks operating under catalysis by proteins through electric fields and organometallic chemistry is what metabolism is. Proton gradients and electric fields drive phosphorylation and are fully integrated into metabolism, and they're transferred through ATP. Metabolism produces coenzymes and building blocks of proteins and RNA. Proteins and RNA and coenzymes. Proteins and coenzymes catalyze metabolism and RNA synthesis, but RNA catalyzes protein synthesis. So we have to explain that. Yeah? And there's the genetic code, which is the association between codons and their amino acid. It's not random, but it's linked to biosynthetic pathways. It appears. And all of this chemistry is compartmentalized into cells that remain always on this 0.1 to 10 micron regime. All of this stuff is important in explaining the origin of life. If we focus on just one thing and ignore all the others, we're missing a huge part of this picture. So now what makes for a good origins of life hypothesis? This is key, because there's many things done in this field. We're studying organics in meteorites. We're studying organic chemical reactions in vastly different environments and with vastly different goals, and usually focus on just one aspect of life. And so what I've been hoping to do in that early part of the lecture is give you a global picture of what life is doing so we can know the things we're trying to explain when we actually try to recreate life in the lab. Okay? And so what I want you to think is that if someone presents you their work on prebiotic chemistry in the future, or you hear about it, it's often in the news, it's a big news story in 5 seconds, you should frame that, you have to interpret that work in the context of all of those things I just showed you. Because if you don't, and it only addresses one thing while ignoring the others, it's not really helping, or only helping in that narrow way, okay? So I think life a good hypothesis should explain all those things that we just discussed in principle. And this is why I wanted to address what I hope I can teach here, or at least make you think about, maybe even if you don't agree with me. When we construct our hypothesis about the origin of life, there's a human tendency to do something I call backloading. Okay? There's a series of things we have to explain. We all know that we have to explain them. But our experiments are designed only to explain one thing. And we do those experiments, and we put in the causal chain of events that need to occur, we put those easy things that are testable up front, and we put the hard things that are not yet testable at the back. And we do the experiments showing that our easy thing works, and we say, hey, look, this shows that this hypothesis, this causal chain of ideas can work. But we haven't shown the hardest things, which are actually the things we need to explain. And then because we haven't shown them, we say, oh, well, evolution invented all those things later, right? So that is, I think, equivalent to this cartoon, which is like you do a derivation, you don't arrive at the thing you're trying to derive, and then you say, then a miracle occurs and derivation is solved, right? So we need to avoid this as much as we can. Of course, we cannot test everything across the entire hypothesis already, but I think our hypothesis from the get-go should address all of those issues and not write off everything to evolution. Okay, so using evolution as an explanation for why things are the way they are is fine, because evolution does occur at some point, but it only occurs if A, there's already a mechanism for evolution that now exists, and usually people invoke evolution without getting anywhere near to that mechanism. B, a transition can be broken down into clear steps, and C, a selection pressure can be identified on which evolution can actually act to give you the outcome that we see in biology today. So I'm not discounting evolution as explanatory mechanism in origins of life, but it can only be invoked under those conditions, in my opinion, for it to be a reasonable explanation. Okay, so let's try to construct our hypothesis in a way that does not do that as much as we can. Okay, so now I'll get a little bit into the history of the field of prebiotic chemistry. And it's not my goal to spend a lot of time on this. I wanted to talk about the famous Miller-Urey experiment that was published in 1953. This was really, I think, a brilliant experiment in many ways, and launched the field of origins of life chemistry and prebiotic chemistry as an experimental research field. And so I think you all already know this experiment. You put some gases into some water into a glass container with an electric discharge, and you let the sparks roll for a little while, and you analyze the products. They're analyzing them using paper chromatography at that time. And they found that here's the actual photo of the chromatography. You can see there's some amino acids that have been detected by certain chemical stains. And so that was a big deal, because now we're making amino acids that life uses through just simple gases and electric charges. This was so important that it ended up in Time magazine, which in the United States is like the biggest thing you could have happen to you probably at that time. That's the same, around the same time that the structure of the genetic code was determined, and this got better headlines than that, even though I think in retrospect that was much more important. And Miller got photographed next to his apparatus for the rest of his life, and continued to work on this and variations thereof for much of his career. He had lots of interesting ideas, actually, that he doesn't really get credited for very much anymore, but a very smart guy. But it kind of, I think, took the focus away from what life is doing, and in fact a lot of the things that life is doing weren't even discovered back then. So there's no way we could even have thought about some of the things I just showed you in the first half of this talk. Probably the next big stage after that was this idea of an RNA world. Now this is a very hard thing to actually talk about, because there's dozens of research groups working on it, each with their own definition of what it is. So I can't really tell you what it is. It's an ensemble of many ideas. But I think one aspect that links them all together, it is a hypothetical stage in the evolutionary history of life on Earth in which self-replicating RNA molecules proliferated before the evolution of DNA and proteins. So this means different things to everybody, but in the purest interpretation, and this is the one I disagree with the most, is that there was no other polymers or metabolism or anything else. All you had was RNA being synthesized by some mechanism that is not metabolism, and that RNA starts to copy itself. And as it copies itself, it might pick up some evolutionary advantage in terms of the copying by catalyzing reactions that end up becoming metabolism. So RNA self-replication ends up producing metabolism and proteins, and then the job of self-replication gets offloaded to DNA. The job of catalysis, because RNA can also do catalysis, gets offloaded to proteins, and that's how we end up at biology as we know it today. This doesn't address things like protein, why ATP, and why proton gradients, and all these other aspects of why metabolism works the way that it does and has the structure that it does. But it gives us this general idea of how we connected metabolism and polymers and the order in which they appeared. That's the most pure version of that hypothesis, really focused just on RNA. There's more kind of open hypotheses that can be as simple as a metabolism already existing and self-replicating RNA emerging from that metabolism. That would be something more like what I would view it as. But I think we should even question the idea, and potentially question, I'm not saying it didn't happen, but we should potentially question the idea that RNA self-replicated at the beginning at all, because that's not how it works today. It's DNA is more of the self-replicator, and RNA replication is enabled by proteins. And so maybe it was the cooperation of proteins and RNA that could help explain how these relationships between RNA catalysis of protein synthesis and protein catalysis of RNA synthesis came to be, right? So let's keep an open mind. This may have happened, but it may not have happened, and let's keep it at that. Okay, so Okay, with that, I'm gonna wrap things up and leave some room for questions. I think we're now at 75 minutes, so let's stop it there. Thank you.